Good morning, friends. And today's presentation we will cover most important kharif crop rice, rice cultivation or package of practices for rice. We know it is a staple food crop of more than 60% of the world's population. It is also staple food crop of more than 70% of the Indian population also. About 90% of the all rice is grown in the world is produced and consumed in the Asian region. Rice contain protein 6 to 7%, fat 2 to 2.5%, amino acids, calcium, and big group of vitamins in low content. The byproducts of rice milling are used as a cattle and poultry feed. Edible oil is extracted from rice bran. Rice husk is used for preparation of cardboard, cement, and insulation material. Rice straw is a good source of cattle feed. Oryza sativa is a botanical name of rice, and this crop comes under Poaceae, that is Gramineae family. Commonly, rice is known as paddy, dhan, bhat. Origin, there are two origins for Asian rice. India, China, and other Southeast Asian countries are considered as origin, while African rice, Oryza glaberrima, West Africa is the origin. As far as geographical distribution is concerned, near about 160 million hectare area is under rice cultivation, and which produces 741 million tons of rice. China, India, Pakistan, Indonesia are the major rice growing countries, contributing about 60% of the world's area. In India, it is grown on area of about 43.90 million hectare with a production of 104.32 million hectare. India ranks first in area and second in production. West Bengal, UP, Punjab are the major rice cultivating states. West Bengal ranks first in area and production. The productivity is highest in Punjab. In Maharashtra, rice grown over and about area of 14.87 lakh hectare with a production of about 42.11 lakh tons. The major rice growing districts in Maharashtra are Thane, Raigad, Ratnagiri, Sindhudurg, Bhandara, Chandrapur, and some part of Nagpur, Kolhapur, Satara, Pune, and Nashik district. Classification of rice. Oryza sativa is a diploid having 24 chromosomes, while Oryza glaberrima is a diploid having 12 chromosomes. The sativa varieties of the world are commonly grouped into three subspecies, namely Indica, Japonica, and Javanica. The information about this species and their characteristics is given below in this table. Here we can see subspecies Indica cultivated in India, Japonica cultivated in Japan, and Javanica cultivated in Indonesia. Indica subspecies is tall in height, which logs easily, having high tillering capacity, awnless or possess short and smooth awns, and grain type is long to medium, while Japonica species having a medium height, not easily lodged, having low tillering, awnless or on, and grain type observed short and round in this Japonica subspecies, while Japonica subspecies having a tall height, not easily lost down, having low tillering on subspecies, and having a large and bold grain type. Now, different growth stages of this rice crop. This rice is divided into two growth phases, having namely vegetative and reproductive phase. Vegetative phase subdivided into three stages, that is seedling, tillering and vegetative lag phase. Seedling stage from germination to fifth leaf stage. Tillering, the plant starts tillering at second week after transplanting and continues up to 7 to 8 weeks. While vegetative lag phase is a sort of rest period, it is a period between maximum tillering and panicle initiation. While reproductive phase, it is a period from panicle initiation to flowering. It consists of 8 substages. Panicle initiation. In this stage, reproductive growth starts. Panicle development. The number of spikelets are decided in this stage. Then booting. The swelling of flag leaf sheath due to developing panicle has been termed as a boot stage. Ear emergence. In this stage, panicle emerge from the flag leaf. Flowering stage. It is also known as blooming stage. Milk stage. The watery content of grain turn into milky consistency. Duff stage. The milky substance later turn into a hard duff with a greenish tint. Maturity stage. In this stage, fully matured grain possess yellow color. Now botany, we will see botany of this rice crop. Botany covers root, stem, leaf and panicle. Roots, like other annuals of Gramini, rice plant has a joined stem, flat leaves and terminal panicles. Roots, after sowing, rice seed gives out seminal roots out of the radicle. These are temporary roots. The real roots are adventitious that are produced from the lower node of the kernel. The root system is fibrous and goes up to 20 cm deep in transplanted rice, while in direct seeded rice more than 20 cm depending upon the availability of water. Stem or kernel, the rice stem is hollow and is made up of series of nodes and internodes. Each node bears a leaf and bud which may grow into a shoot or tiller. Tillering continues during the vegetative stage, that is up to 60 days after transplanting. Some of these tillers are died during the reproductive phase due to competition for water and nutrients. Leaf, the leaf of rice is divided into leaf blade and leaf sheath. In most of the rice varieties, the leaf sheath may almost completely cover the upper internode. Now panicle, the inflorescence of rice is known as panicle. Rice panicle is branched and branches bear spikelets. Rice is a self-pollinated crop. Rice fruit is a single seed fused with wall of ovary and known as caryopsis. Now climate. Rice is a short day thermosensitive plant. It requires hot and humid climate. The regions having high humidity, prolonged sunshine and assured supply of water are best suited for rice cultivation. The average temperature required throughout the life period ranges from 21 to 35 degrees centigrade. The level of solar radiation determines the productivity of rice. Inadequate solar radiation during reproductive stage and ripening stage reduce the number of spikelets and percentage of field spikelets. Now we will see soil. Rice is a semi-aquatic crop grown on a wide variety of soil. Soil should be capable of holding water for a longer period, such as heavy soil are more suited for its cultivation. Rice grown successfully on alluvial, red, lateritic and black soils. pH of this soil should between 5.5 to 6.5, and being semi-aquatic crop, it grows well under submerged condition. Now different systems of rice cultivation are there, and there are main four systems of rice cultivation which are followed in India, which are as follow. First one, dry or semi-dry system, rain-fed upland cultivation. Wet system, lowland cultivation. Direct seeding of sprouted seed, and system of rice intensification, that is SRI method. Now dry or semi-dry, we will see one by one in detail. Now dry or semi-dry system or rain-fed upland situation. Actually, this dry system of rice cultivation is followed in uplands. Uplands, we know, uplands are characterized by aerobic soil in which attempt is made to impound water. Uh, Dry system is followed in the areas which receive annual rainfall 800 to 1000 mm and there is no irrigation facilities. Under such a situation, it is better to grow short duration varieties. Most of the part of UP, Bihar, Orissa, Tamil Nadu and Andhra Pradesh is under rain-fed upland condition. In Maharashtra, this system is followed in Usmanabad, Nanded and some part of eastern districts of Maharashtra. And in semi-dry system, rainwater is impounded in the field after completion of weeding, interculture and fertilizer application when the crop is about 1.5 to 2 month old. This type of cultivation is followed in Assam, West Bengal, Eastern MP, some parts of Orissa and Eastern Maharashtra, that is Chandrapur and Bhandara districts. Now land preparation for this dry or semi-dry system. Plow the field immediately after harvest of the previous crop, or during summer season we have to plow the field. Then carry 2 to 3 crisscross harrowings with monsoon arrival, and broadcast or apply FYM uh, before last harrowing, so that that FYM will get thoroughly mixed in the soil due to last harrowing. Then bunch should be prepared around the field to check the loss of rainwater through runoff. Then seed and sowing, generally first fortnight of June to first fortnight of July is sowing time. In some part of the country, sowing is carried out before the onset of monsoon, that is last week of May. Seed treatment, seeds should be treated with 3% brine solution to prevent the crop from diseases. The floating seeds are removed and seeds settled at the bottom are used for sowing. Then seed treatment with thiram at the rate of 2.5 to 3 gram per kg should be done to control blast, brown leaf spot and sheath blight disease. Then methods of sowing, broadcasting and drilling. Then seed rate for broadcasting 100 kg seeds per hectare in this spacing not maintained, and in drilling 60 kg per hectare seeds are drilled with 20 centimeter row to row spacing. Then nutrient management, apply 10 to 15 ton FYM or compost per hectare before last harrowing. The recommended dose of fertilizer is 120-60-60 kg NPK per hectare. 50% of nitrogen and full dose of phosphorus and potash should be applied as basal dose, while remaining 50% nitrogen should be given in 2 equal splits during tillering and panicle initiation stage. Application of zinc sulfate at the rate of 25 kg per hectare found beneficial to correct zinc deficiency, while foliar spray of 0.5% ferrous sulfate effective against iron deficiency. Weed management in a semi dry system or dry system. Weed competition is more severe in direct seeded rice. Period of 30 to 40 days most critical for weed competition. Two hayings should be followed by one hand weeding will help to control the weeds effectively. Then we can go for herbicide application also. Pendimethalin stomp at the rate of 1 to 1.5 kg per hectare as a pre-emergence, or oxidized 0.120 kg per hectare to control weeds effectively. Now second system of cultivation, that is wet or lowland system. Wet or lowland system of cultivation is followed where adequate irrigation facility is available. The field is brought to a soft condition by puddling and sprouted seeds are directly sown or seedlings are transplanted in the puddled field. Preparation of nursery. Nursery is prepared on well-drained fallow land near the source of irrigation. In general, 1/10 of the nursery area is sufficient for seedlings to transplant 1 hectare area. Now, seed rate for 40 to 50 kg per hectare. Sorry, the nursery seed rate 40 to 50 kg seeds per hectare. Nursery prepared by three methods: wet nursery, dry nursery, dapog nursery. Now we will see one by one. Wet nursery. Wet nurseries are preferred under irrigated condition. Nursery land should be plowed in dry condition and again puddled by plowing 3 to 4 times. Then 3 to 4 times in 5 to 6 centimeter standing water. Then apply FYM or compost at the rate of 10 to 15 tons per hectare at the time of first puddling. Field should be leveled after final puddling. Then after 1 to 2 days, prepare narrow beds of 120 to 125 centimeter width or and of any convenient length depending upon type of soil. Construct the drainage channel of 30 centimeter wide in between beds to facilitate drainage. Then apply 0.5 kg N, 0.5 kg P2O5 and 0.5 kg K2O per 100 meter square area or 1 R area before final leveling of the nursery beds. Then broadcast 2 to 3 handfuls of dry or sprouted seeds per square meter on that seed bed. For sprouting, healthy seeds should be soaked for 24 hours in clean water. Then the seeds are kept in moistened gunny bags for about 24 to 36 hours. Then seedbed should be saturated with water up to 5 days. Then gradually increase the level of water up to 5 centimeter. Then drain out excess water in case of heavy rains during first week of sowing. Top dressing with 1 kg urea per gunta or per 100 meter area should be done after 15 days of the sowing. Seedling become ready for transplanting at the age of 20 to 25 days. Now we will see information about dry nursery. This nursery followed in the areas where non-assured water supply is there. Land selected for dry nursery should be moisture retentive. Plow and harrow the land for good tilth. Then apply FYM or compost at the rate of 10 to 15 tons per hectare and mix thoroughly in the soil. Then prepare raised bed of convenient width and length with a top of 10 to 15 centimeter in height and of any convenient length. Then 30 centimeter wide channel should be kept in between two beds to facilitate drainage. Then apply 0.5 kg N, 0.5 kg P2O5 and 0.5 kg K2O per gunta or per 100 meter square area. Sowing should be carried out at the depth of 2 cm in rows at 8 to 10 cm apart. Cover these seeds with a thin layer of soil immediately after sowing. Then special care should be taken for water management in these beds. Allow the water to run in the channel first and then raise level of water slowly to saturate the soils of that bed. Apply 1 kg urea per 100 meter square area as a top dressing 15 days after sowing, and within, in this case also, within a month or 20 to 25 days, seedling become ready for transplanting. Then next one, dapog method. This method actually introduced in India from Philippines. In this method, nursery is sown on concrete floor or on raised bed of soil covered with polythene sheets or banana leaves. This method is adopted in areas of assured water supply and when early transplanting is to be done. A small area of 25 to 30 meter square is sufficient to raise seedling for 1 hectare area. Pre-germinated seeds are broadcasted uniformly over the polythene sheet or banana leaves at the rate of 3 kg seeds per meter square. Then sprinkle water over the seeds 3 to 4 times in a day till 4th day to keep the seed bed wet. Press the seeds gently with hand or with wooden flat board 1 to 2 times a day till first 3 days so that roots remain contact with the water. When the seedling attains 2 cm height, a constant film of water should be maintained. Within 12 to 14 days, seedling become ready for transplanting. Seedlings are very delicate in this case. The nursery is cut into strips and rolled like a mat with roots outward. Loose the interlocked roots carefully before transplanting and transplant 3 to 4 seedlings per hill in dapog method. Now we will see advantages of this dapog method. It requires half of the time for raising the seedling. Even area also required less for nursery. Seedbed preparation doesn't require. Labor cost of raising nursery is very less as compared to other methods, reducing crop growth period by 7 to 10 days. There are some disadvantages also of this dapog method. Seedlings are very delicate, thin and slender, and seed rate is high in this case. Now field preparation. Plow the field immediately after harvest of the previous crop. Summer plowing, as we know the importance of summer plowing, which help in exposing the eggs and larvae of harmful insect, pests and rhizomes of the weeds. Then stubbles, weeds and other plant remains should be picked up and thrown out of the field. Then bunds should be prepared around the field before puddling and apply 10 to 15 ton FYM compost per hectare before last harrowing. Puddling, keep the field flooded or saturated with water of 15 days before transplanting. Puddling is done by running the recipro or puddler, either bullock or power drawn, in standing water. Puddling helps to retain water in the same field due to closing of micro and macro pores of the soil. Then transplanting. Age of seedling. At the time of transplanting, seedling should have 5 to 6 leaves and it should attain a height of 12 to 15 centimeter. In Kharif season, 21 to 25 days old seedlings, and in Rabi season, 30 to 35 days old seedlings are ideal for transplanting. Time of transplanting. Second fortnight of June to first fortnight of July is a right time for transplanting of rice in Kharif season. While for Rabi season, last week of December to middle of January is the optimum time for transplanting. Spacing 20 centimeter by 15 centimeter or 15 by 15 centimeter is the ideal spacing. 2 to 3 seedlings are transplanted per hill to a depth of 2 to 3 centimeter. If seedlings are overaged, then 4 to 6 seedlings per hill are planted with closer spacing. Third method, broadcasting of sprouted seeds. This method is followed in areas where there is a shortage of laborers and labor wages are high. The field is prepared and puddled in the same manner as that for transplanting. The seeds are soaked in water and kept at room temperature for 24 hours. Remove the seed from the water and put it in a clean and moist bag. Fold the bag tightly and keep it in a shade for 24 to 36 hours. After every 12 hours, store the seeds for air circulation and sprinkle the water on seed. During this period, the seeds get sprouted. Then the sprouted seeds with a radical length of 1 to 2 mm are broadcasted on a puddled field at the rate of 100 kg seeds per hectare. Manures and fertilizers, add 10 to 15 ton FYM or compost per hectare before last harrowing, and recommended dose of fertilizer is same 120 60 60 kg NPK per hectare. Here also 50 kg nitrogen, 50% nitrogen and uh, full phosphorus and potash should be given at the time of, sorry, not 50%, 40% nitrogen and full dose of phosphorus and potash should be given at the time of puddling, and remaining 40% nitrogen is given in two equals is given at tillering stage and 20% at panicle initiation stage. In case of zinc deficient and ferrous deficient soil, apply zinc sulfate at the rate of 25 kg per hectare, and 0.5% take a foliar spray of ferrous sulfate 0.5% to recover the deficiency, boron deficiency, iron deficiency. And for boron deficiency we can spray 0.2% borax. Green manuring in situ or green leaf manuring get a importance in the recent years. Incorporation of glyceria at the rate of 10 ton per hectare at the time of puddling proved better for increasing rice yield. Biofertilizers such as blue-green algae azolla are also capable of providing 20 to 25 kg nitrogen per hectare. Now, water management. Until the transplanted seedlings are well established, water should be allowed to stand in the field at a depth of 2 to 3 centimeter. Thereafter, about 5 centimeter level of water may be maintained up to the duff stage of the crop. Then water should be drained out from the field 7 to 15 days before harvesting, depending on the soil, to encourage quick and uniform maturity. Interculturing. The intercultural operations carry out 30 days after transplanting with a rotary or Karjat or Japanese hoe to control the weeds. Crop rotation. In irrigated areas, rice groundnut, rice cowpea, rice horse gram, rice wheat, rice potato, black gram, and so on. Different cropping systems are there. In unirrigated areas, rice field bean, rice field pea, rice chickpea, rice lentil. These are the some crop rotations. Now last one, system of rice intensification, that is a SRI method. System of rice intensification is developed in Madagascar in 1980s. Before that, up to 1997, this technique not known to the world. The average yield obtained in SRI is higher than the conventional transplanting method. SRI method practice produce more tillers with a positive correlation between tillering and grain filling. The plant has deeper and more effective root system due to wider spacing. Some advantages or merits of SRI method are there over a conventional method. Water requirement is less. Seed requirement is 5 kg per hectare, which reduces the cost of seed. Yield is more than conventional method. Crop mature earlier than conventional method by 7 to 10 days earlier. More number of grains per panicle than conventional method. Now nursery management, for this SRI method, mat nursery is prepared. Raised bed 120 centimeter wide are prepared with a 100 gauge polythene sheet at the base by making holes for drainage. Apply 2.5 centimeter layer soil and FY mixture in 2:1 proportion. Distribute the sprouted seeds at the rate of 50 gram per meter square for fine grain varieties and 100 gram per meter square for coarse grain varieties uniformly on the seed bed, and cover it with another thin layer of soil and FY mixture. Then cover the seed bed with paddy straw and watering done regularly to keep the seed bed moist. Field preparation, field prepared similar to that of conventional planting. Transplanting about 11 to 12, 14 days old rice seedlings used for transplanting. Seedling Single seedling is planted at 25 by 25 centimeter. The seedling should not be planted too deep into the soil. Use of organic manure incorporate well decomposed FYM or compost at the rate of 10 ton per hectare. Then irrigation management. Regular irrigation is necessary to keep the soil moist. At early growth stage, flooding of the field should be avoided. Water should not be allowed to stagnate at any stage. That is the main importance in this SRI method. Intermittent wetting and drying for adequate aeration is essential. Weed management. Weed infestation is severe in this method. Hoeing with rotary weeder or cone weeder is effective to control the weeds. Hoeing should be done in both directions starting from 10 days after transplanting. Because of wider spacing, we can go for weeding in both direction, hoeing in both direction. About four such hoeings are given at 10 to 15 days interval. One or two hand weeding may be carried out to remove the weeds closer to rice plants. The varieties, there is a group of early, late, mid-late, late, and hybrid varieties. This is given in this slide with days. Early duration require 100 to 100 to 110 days. Mid-late duration requires 120 to 130 days. Late duration require 140 to 145 days, or hybrids require 120 days. Now harvesting and threshing. Crop should be harvested at 80% physiological maturity stage. Ears are nearly ripe and the straw is still slightly green. Crop generally become ready for harvesting 25 to 30 days after flowering. When 90% grains in panicle mature, harvesting should not be delayed. Timely harvesting ensures good grain quality as less grains are broken during milling. Harvesting carried out by manual labors. Plants should be cut close to the ground level and dried in the field for 2 to 3 days. Later on, taken to the threshing yard for threshing. Most common method of threshing is striking bundles on the raised wooden platform, trampling by bullocks, rubbing with bare human feet. Nowadays, pedal or power driven threshers are used for threshing. Harvested grains should be dried under shade to bring down the moisture content to around 14% for a storing and milling. Yield from early varieties we will get 40 to 45 quintal per hectare grain yield. From mid-late and late varieties 45 to 55 quintal per hectare yield, and from hybrid varieties we will get 60 to 70 quintal yield, grain yield. Now insect pest management. Stem borer is one of the important pest which damage the larva of this pest, which results in dead heads or white head larvae bore and feed inside the stem. To control this stem borer, destruction of stubbles previous crop, grow tolerant varieties like Ratna RP414, IR20, IET 3116, IET 3117. Seedling root dipping in 0.05% chloropyrifos solution before transplanting. Apply carbofuran at the rate of 0.5 kg per hectare or phosphate at the rate of 10 kg per hectare in 3 to 4 centimeter standing water in field 20 to 25 days and again 50 to 60 days after transplanting. Second one, gall midge. To control this gall midge, sowing tolerant varieties like Phalguna, Vikram, Pratap, IET 6080. Destroy affected tillers and apply carbofuran at the rate of 16.5 kg per hectare or phosphate at the rate of 10 kg per hectare in 3 to 4 centimeter standing water in the field. Then brown plant hopper, green leaf hopper. To control these hoppers, so resistant varieties IET 7575, IET 6315, MTU 4870 Triveni for brown plant hopper, and Vijaya IR20 IET 7301 for green leaf hopper. Avoid close spacing. Avoid excess use of nitrogenous fertilizer. And if hoppers are observed, then spray monocrotophos 0.05% or malathion 50 EC 0.1%. Then Gundi bug or earhead bug. To control this pest, clean cultivation, destruction of the grasses, and apply malathion dust at the rate of 20 kg per hectare. Then leaf folder. To control leaf folder, spray 0.15% malathion or 0.15% carbaryl or 0.05% monocrotophos. Case worm. To control case worm, remove and destroy tubular cases. Spray 0.05% monocrotophos. Now last, disease management. Reduction in yield due to various diseases have been estimated about 10 to 15%. Blast is one of the important disease. To control varieties like Ratnagiri 24, Karjat 1, IR8, and seeds should be treated with thiram at the rate of 3 gram per kg of the seed. Foliar spray of tricyclazole 0.01% or copper oxychloride 0.3%. Brown lip spot. To control this disease, seeds should be treated with thiram at the rate of 3 gram per kg of the seed. Spraying of copper oxychloride 0.25%. Then a false smut. To control false smut, sowing resistant varieties like IR20 to 26, IR28, Vijaya, and spray of propiconazole 0.1%. Bacterial leaf blight should be controlled by seed soaking for 12 hours followed by hot water treatment at 52 to 54 degrees centigrade for 30 minutes. Sowing resistant varieties and spray of copper oxychloride 0.25%. All alternately with streptocycline to 50 ppm. Last one, tongro disease, sowing resistant varieties IR50 and Co 45, and control leaf offer are the control measures of this tongro disease. In this way, in short, we cover this rice crop. Now references are given here as usual: Principles and Practices of Agronomy by Balsubramanian, Modern Techniques of Raising Field Crops by Chida Singh, Handbook of Agriculture by ICAR, Textbook of Field Crop Production by Rajendra Prasad. Thank you one and all friends for watching this presentation. Kindly I request all of you to watch, share, like and subscribe this channel page. Thanks once again.